How do you guys feel about flipping on these lights? Favorable? Okay, cool. I feel like we're just gonna go to sleep otherwise. I know. I prefer low light most of the time, but it's just too sleepy. And this stuff that can be a little bit on the dry side, admittedly. All right, so I didn't prepare any kind of review questions. I usually do, but there is some kind of built-in review as we start ramping up on T-cell activation, so hopefully your memory will be jogged on some of this. I also, when I was going through these slides for today, identified a few mistakes, so I corrected those out and annotated on the slides. So make sure, like, whatever you're doing to take notes, make sure you're actively taking notes, and if I catch a typo or a ridiculous sentence that is out of place, I'll put it on this slide and draw your attention to it so you can also do that on your notes as well. So I went through this deck fast, and there are already a lot of changes I'm making. Okay, so we'll keep it mostly restricted to this today. It's a little out of order, like we're going to be talking about what makes the adaptive immune system the adaptive immune system, even though we've talked about before. We'll start by talking about that again. Then we'll talk about those primary and secondary organs of the immune system. And then we'll get into some really nitty-gritty specifics regarding T-cell activation and memory. But tomorrow, that's when we'll hit B cells and their functions. Or, not tomorrow, Monday. Monday at 1500, right? It's a brutal slide. Okay. That's gonna suck. Exactly. Okay, so the most important, if we're talking about general characteristics of lymphocytes, both B cells and T cells, these adaptive immune cells, the most important one to remember is this one, this bolded antigen-specific receptors. This is what contrasts these cells with your innate immune cells. They are, at this point, recognizing specific parts of those antigens called epitopes. So we'll get into that a little bit more later as well, but what this diagram is showing you that on B cells we have our B cell receptor, our BCRs, and if you recognize that Y-shaped molecule, that's pretty much an antibody. It's a membrane-bound antibody, that's what B cell receptor is. On the T cells, we have a TCR, our T cell receptor. Very similar structure in a lot of ways. It's got its transmembrane domains, an alpha and beta chain, which you don't need to know about too much. Then on the surface side, we have a constant region, and then we have variable regions for both of these molecules. In this cartoon, it's a little confusing because the T cell receptor is actually shown docking with the MHC complex of an APC. So on this side, you would have an antigen-presenting cell with its MHC presenting this antigen, the epitope on the antigen, to a TCR. The cognate for the B cell receptor would be similar. You would also have an epitope. But in this case, you have two binding domains on that antibody. So you have, these are called FABs, we'll talk about that more later, but they're basically two, they're bivalent. So one can recognize one antigen, the other can recognize another. Okay. In terms of diversity, these guys are generated with a specific, yes sir?
I just have only slightly related question. So you said, is it the inner portion or the outer portion of the atoms?
Outer side.
Okay.
So this is on the inside of the cell, and then on the outside, these are what's sticking out for all the world to see to interact.
So you said it's a fab. Is that where we get derivatives of medications like Anivil, like Profound?
Say again?
Is that analogous, or is that like where we get the derivative for medications like Profound, which is a protein?
We do get some countermeasures from antibodies, like monoclonal antibodies, that are designed to have specific antigen receptors on their FABs. But I don't know if that's what you're talking about. Wait till next lecture, and if it doesn't like, if you don't get a yes or no in your mind during that lecture, let me know and I'll look it up. But yes, we do have the capability of synthesizing therapeutics that take advantage of that specificity. Okay, so
Question, ma'am.
yes.
With the fork has like four receptors, does that mean that we can find 4 antigens on it?
That's a great question, and no, it means we combine 2. So this is a little misleading, but this is for one antigen and then another. diversity where we could recognize virtually any type of epitope imaginable. And again, those epitopes are defined as small amino acids, 18 to 30 amino acid long peptides that they can recognize. That would require some sort of background in eukaryotic genetics and how those B cell and T cell receptor cassettes are organized in the genome. But if you've ever had a class in this before, you know it's like staggering. Has anybody ever studied like VDJ recombinatorial cassettes before? Does that ring a bell? Okay. Well, suffice to say, like, it is way in excess of what you would ever need, the amount of diversity that can be generated with these receptors. So what comes out of these hematopoiesis processes when these cells are starting to differentiate and they're starting to express different BCRs and TCRs is, I'm trying to think of a number that's big enough. So we're talking about 10 to the 20th or so that's like over a quintillion possibilities of different antigens you can express. So that's what we're going to talk about a little bit today. We're not going to talk about the genetic part of that because that would be crazy, but there's a lot of somatic hypermutation involved, there's a lot of rearrangement, there's a lot of mRNA splicing. It's very complex, but you get a lot of diversity, way more than you need, okay? And you're born that way, and if you're never used, you're never used. But if you are needed, you're already ready to go. That's the whole principle behind ready-made specificity. Two other characteristics that are important to remember are one, both of these cells, B cells and T cells, are long-lived, and if they do meet the right antigen, they are selected for and they start expanding. That's clonal expansion of that exact right, correct epitope recognition receptor. And then immune tolerance, which I'll circle back to in a second. So this figure that I chose to complement the text here is with B cells, but T cells go through a similar selection process. And just like what I was saying verbally a second ago, when you generate these B cells and T cells and they have their specific, antigen-specific TCRs and BCRs on their surface, you will have cell 1 with its specific BCR, cell 2 with its different specific BCR, cell 3, 4, 5, whatever, all the way up to like 10 million. So you're talking about a population in the lymph node, and there's about 10 million of those in there. Okay, so most of those aren't going to do anything. It's kind of analogous to like a fighting force, right? We're all wearing this uniform, we're theoretically ready to go engage and do whatever we have to do. But in reality, most of us don't actually do that. There's a select few that ever get activated to do that. So that would be kind of like what's happening with number 2 here. This is designed to recognize one antigen. It meets that one antigen in a lymph node, probably. And in that lymph node, once it meets that antigen, a whole bunch of signaling cascades get activated, which tell that one particular B cell to expand, to make more of itself. So there's a lot of cytokines that are going around signaling. But at the end of the day, what happens is you get massive proliferation of that one specific B cell. Same with T cells, very analogous. Types of differentiation here, further differentiation, until you get this effector cell that's called a plasma cell. Plasma B cells are the ones that are actually now secreting antibody. They're what you think of when you think of like antibody secreting cells. So you're making a whole bunch of antibody, and all that antibody, the soluble protein that's going all throughout the circulation, is going to be really good at binding to those specific antigens and helping you to clear that very effectively. That's what that memory is for. At the end of this little engagement, you're either dead or you live, right? And if you live, you clear the pathogen. So there's no more threat. You don't need all these expanded clonal populations anymore. Most of them will start dying off. They'll undergo apoptosis just through lack of attention. It's called an anergy. They'll start committing suicide. However, a select few will go on. These are called memory B cells or memory T cells, either one. And those are the ones that will persist for years, like possibly for the rest of your life. And they're also the ones that are going to be responsible for fighting that same pathogen again, should you ever encounter the same exact pathogen again. Okay. Yes, sir.
Is this what comes into play whenever they're talking about donating blood and stuff like that? Is because I get emails all the time saying like, oh, you produce this antibody, like we want you to donate blood.
Well, with blood donation, there's a couple things. So one, we screen our blood supply for different pathogens, bloodborne pathogens. Two, there's a compatibility issue, and I think that's what you're getting at, you know, if you have like, if you're O positive, A positive, B positive, those are antigens that are on the surface of erythrocytes, right? So that's a kind of a different thing than what we're talking about there. But your immune system needs to be comfortable in its own body, right? So that's another thing we're gonna be talking about. It's not designed to be accepting towards other people's tissues and fluids. So if you get a transfusion and you're not matched with that person, your immune system is designed to appropriately freak out. That's why we match donors. Okay, speaking of, now getting into organs of the immune system, we have two primary legit organs, the bone marrow and the thymus. We've talked about this a couple times, that all blood cells, white and red alike, are born, if you will, in bone marrow. So with the white blood cells that are in the bone marrow, this is also where our lymphocyte populations are generated. These are very badly phrased bullets, but both B cells and T cells are produced in the bone marrow. Okay. T cells are a little bit more complicated in terms of their maturation processes. So shortly after their precursors or progenitor cells are produced in the bone marrow, they have to migrate from the bone marrow to the thymus. Where is the thymus located? Yeah, right, kind of in the middle of the chest, between the lungs, the mediastinum. Yeah, tiny little organ, and it only gets smaller over time, right? It just shrinks and shrinks and shrinks and becomes increasingly fatty. But that, nonetheless, throughout your life is where those T cells go as they're generated in the bone marrow. It's kind of like IPAP in a way. Like phase 1 is going to be in the bone marrow, and you get what you can get out of it in terms of maturation and progression. But at a certain point in time, you need to go to phase 2 and get an entirely different learning environment, more clinically relevant learning environment once you're ready. Same with T cells. They get what they can get out of the bone marrow, but once they go to the thymus, they are exposed to like this, it's like an entire training system for these T cells. The body has specialized, almost like mock epithelial cells that train them what to leave alone and what to target. It's very, very cool. It's like obedience school for your dog, you know, don't chew on the $1,000 Balenciagas or Louboutins, whatever. You know, training these T cells. They need that. They need, because of their specific role, because of why it's different than B cells, they need to undergo positive selection, where they learn to recognize their own tissues and cells and they don't attack those. And they undergo negative selection when they are too aggressive or overly aggressive. Okay. And then at that point, they will finally differentiate. So up to that point, they don't have those markers of what they will finally become, which is either CD4 helper cells or CD8 cytotoxic cells. Okay. In terms of secondary lymphoid organs, this is where it gets a little bit complicated, but we have our lymph nodes, which are super important, and then our spleen. And if you want to consider this an organ, great, but it's more of a tissue. We talked about the mucosa of our body and how important that is in the immune system. So all these MALTs, all these mucosa-associated lymphoid tissues have really, really important roles as well in the immune response. But briefly, going back up to your lymph nodes and what these things do, let's start with what lymph does. What does lymph do for the body? It's lymphatic fluid. Yeah, exactly. All that extra fluid needs to be filtered and pumped through your lymphatic system, which will filter all out during the process. It'll filter out all the antigens and bad things that it encounters, both foreign microbial as well as cellular junk as well. So all that will go through the lymph nodes. And if you look at this figure, the reason why I swapped out figures, this one is nice because it says nice and cleanly primary lymphoid organs versus secondary lymphoid organs. This one is nice because it shows you the sheer number of lymph nodes that you have. You have, depending on the person and their age, probably between 500 to 700 different lymph nodes in the body. These are kind of like, your body is like an entire battlefield. These are sort of like tactical operation centers where where there's a lot of immune decisions being made at each one of those lymph nodes. So pretty complicated. They are concentrated where? Where do you have the most lymph nodes in your body? Cervical, axillary, in the armpits, and inguinal, in the groin. You have tons and tons. So when you get sick, especially cervical lymph nodes, you know, if you've ever felt those kind of swell up and you have some lymphadenopathy, those are lymph nodes that are having an influx of immune cells and immune response is being mounted at that time. So that's what those lymph nodes do. With the spleen does a bunch of things. It's a multifunctional organ. That sits kind of behind the stomach, up here in the upper left quadrant of your torso. And it's sort of like the lymph node for the blood. So your spleen is going to filter out all blood, all bloodborne pathogens, and that will also play an important role. Patients who are splenectomized or asplenic generally have a tough time fighting off disease. You know, it's not a big disadvantage there. And then finally, again, like with the malt, what were some of those areas in our body that we said had like mucosal areas? Nasal, mouth, our entire GI tract, right? It's one big tube, so it's like all mucosal from start to finish, and the urogenital tract. So all of that is mucosal tissue. All of that has its like, we have our niche names, the nault, the nasal associated, the balt, bronchus associated, and the galt, gut associated. Probably some others as well, but those are some of the major ones. And the Waldeyer ring shows up here. That's like the tonsils and adenoids around the pharynx. So I chose one to kind of drill down and get a little bit more specific as an example because we do study enteric pathogens so much in block 3, so I figured this would be a good one to kind of prime you on how one of these major lymph nodes work. This major lymph node is a mesenteric lymph node that is situated in the mesentery, which if you think of your intestines as like a garden hose, the mesentery would be the thing that you use to like wrap the garden hose around and keep it nice and tight in situ. So with it, in terms of how this is arranged writ large, you guys haven't started talking about GI at all, EMP. Okay, so this cartoon basically is showing you, just to orient you, you have the lumen of your intestines, which is where all the food is going through, and of course that's where all your gut bacteria are as well. So we've got a couple of bacteria up here. We have a barrier functioning with this mucosal epithelial layer, separating out the gut from just like a nice tight barrier so your gut contents don't leak into the rest of your body. The lamina propria, that is a connective tissue that is packed full of different immune cells, that's what we'll go over in just a second. And then that mesentery, of course, which is like the organizational part. So their peritoneal cavity, that mesentery would be like the outside of that peritoneal cavity. Okay, so what is going on here? Somebody asked a really good question yesterday or the day before about how we don't go berserk when we have all these gut bacteria that we have to sift through and make sure that they're friendly and they're not trying to hurt us. Because we do have tons of legit enteric pathogens that cause problems for us. We have Salmonella, Campylobacter, E. coli. So these cells have to be specially trained to separate out the trillions of friendly commensals from the very select few pathogenic enteric bacteria that actually cause problems for us. And one of the ways they do it are they have some specialized follicles under the mucosal epithelial cells, these follicles that have Peyer's patches in them. And Peyer's patches are just part of that epithelial barrier that are enriched with things like M cells, or microfilled cells. They have this specialized architecture that allows them to capture antigens from the lumen of the intestine and transcytose them across down into the lamina propria where they can present them to all these immune cells there. We've also got our old favorite, the dendritic cell. What's that best at doing? Dendritic cells? Antigen presentation, so it does what it does, right? It sticks its little tendril up through those tight junctions in the epithelial layer, and it starts sampling with its little dendrites, and then it pulls those antigens back down. It does the same thing as an M cell. It presents that to other cells and shares that information. Goblet cells, anybody know what goblet cells do? yeah, they make all that mucus, and that mucus has an important function too. I mean, that's why it's the mucosa, right? It's covered with mucus. Panath cells, Panath cells, guess what that is? Those are responsible for, among other things, they secrete a lot of antimicrobial peptides, an important part of the innate immune system. So yeah, we have a lot of sampling going on. When these APCs are picking up antigens of interest, they are doing what they do, they share it. So here we have our lymphatics, afferent lymphatic system. So here we have a dendritic cell, it looks like it's carrying something of interest. It's gotta carry that down into this TOC, this tactical operations center, right? This mesenteric lymph node, and it's gotta share it with T cells. And those T cells then become activated if they're the right T cell. And then they will migrate back up to this problem area and solve that problem. Same with B cells. Activated B cells will come and be recruited to solve whatever problems are there. So, also you do have some plasma cells. These are, again, differentiated B cells that are now secreting specialized different types of antibody. And here you see a type of antibody we haven't talked about, any antibodies in detail, but this is IgA, secretory IgA, a dimer that's really, really important for all these mucosal surfaces. So this plasma cell is secreting secretory IgAs, which can translocate across mucosal epithelial cells, that barrier, and go into the lumen and handle some of those specific pathogen threats that way. A bunch of different mechanisms coming together. Okay, any questions on that example, lymph node? Yes.
So just to make sure I'm reading this diagram correctly, read from left to right.
This one,
Yes.
left to right? I think if the story makes sense, you start right to left, top to bottom, like however you want to read it. Yeah. Maybe the way to do it is top down, when you're starting from the lumen and you make your way all the way to that communication center in the mesenteric lymph node. That's the MLN, mesenteric lymph node. Yeah. But again, like these figures, they're not meant to confuse you, they're meant to kind of show you how this is all coming together. So, if it doesn't make sense, you know, just let me know. Okay, so in terms of surface markers, these lymphocytes, B cells and T cells both have a bunch of surface markers when they're differentiated fully, even when they're undifferentiated. Those surface markers give you an indication of what their effector function is, and it has a role too in how they interact with other cells. So we'll spend a couple slides talking about some specific and really, really important surface markers for lymphocytes. But those effector functions ultimately result in things like signal transduction, cytokine secretion, things like that. And some of them play a role in adhesion. So for T cells, one of the most important ones, since an indicator of mature T cells, is I forgot to tell you what CD stands for. We've been talking about CD4 and CD8. CD stands for clusters of differentiation. It's an old term, it's been used for a long time. In the field of immunology, unfortunately, a lot of these surface markers that have been described, there are hundreds of them, have been given double names. So I'll try to use CD whenever possible, but just know that a lot of them do have double names. Okay, so clusters of differentiation, number 3, 4, 8, etc. With number 3, this is an important marker on all mature T cells. You need it in order to have effective signaling through the T cell receptor. Then you have the primary molecule, which isn't on here, that's like the T cell receptor itself, right? The T cell receptor is binding to what on the antigen presenting cell? MHC. Yeah, MHC. You guys remember talking about major histocompatibility? Okay, so if you are cytotoxic T cell, CD8 cell, which one of those MHC complexes are you binding to? If you're a CD8 cell, you're binding to 1, MHC1. And if you are a helper cell, a CD4 cell, you're binding to MHC2. Yes, okay. So these are co-receptors. So you have that primary interaction between the T cell receptor and the MHC complex, but then you also have to have one of these co-receptor. That's what it is if you've been wondering what CD4 and CD8 is. It's a co-receptor. And you'll see that in the next diagram in a second here. In terms of relative abundance, there are a lot more CD4 cells than CD8 cells. And also note that you don't, for mature functioning T cells, you don't have both. You have one or the other, separate functions. You also have a ton of other ones, but importantly, CD28 is a co-signal. It binds to another important marker on antigen-presenting cells called CD80, or more commonly B7, it's a more common name. And then I mentioned that some of these surface markers are also important for adhesion, like LFA1, leukocyte function associated antigen. LFA1 is super important. So those are some of the key surface markers for lymphocytes. For B cells, the B cell receptor is what again? Like what kind of molecule is that on the B cell? It's an antibody. It's a membrane-bound antibody. That's the B cell receptor. So the cognate is MHC2. You have part of that B cell receptor complex is CD19, CD20, and CD21 all together. But CD19 is the one we use as a marker for mature B cells. And then you also have this other other surface marker called CD40. And that provides a co-stimulatory signal for binding with T cells, B cells that bind with T cells together. Right. Looking at the way some of these interactions might occur temporally, we have our antigen-presenting cell at the top of all four of these different cartoons that are laid side by side. So our antigen-presenting cell is up here, and it's showing us 4 different examples of these binding interactions that occur with different surface markers. So here's our T cell on the bottom, APC's on the top. The first one is that adhesion interaction that's going on between LFA1 on the T cell and an integrin called ICAM that would be on the surface of your APC. Next, we have, here we actually see either our CD8 or CD4 co-receptor finally. So we have our T cell receptor, that's this reddish shape that's coming up. It is receiving this peptide that's being presented on either MHC1 or MHC2. And then CD8 or CD4, one or the other, but not both, is extending up as a co-signaling molecule. So you see how it's touching the end of MHC1. That touch is making sure that it is self, that that's the right signal. Without that, you don't get antigen presentation. It doesn't work without that important CD4, CD8 interaction that happens there. After that, we need a co-stimulation interaction. So here we have CD8, which was on this last slide. CD8 receives activation co-signal and binds to CD80 or B7. So here we see B7, it's binding to that. That's that, kick in the pants co-stimulating function that says this is, you need to take action. This is confirmed. This is a confirmed interaction. And then finally, we have cytokine receptors as well. So cytokines are still involved. They're still needed for appropriate signaling. Some of those important cytokines we've talked about are IL-1 and IL-2. We need those as well. So they have their cognate receptors on those T cells. And you can see, we talked about IL-2 before. This is an important cytokine for T cell proliferation. And what this little arrow coming up from the T cell is showing you that not only can you receive those signals from neighboring molecules, which would be like a paracrine type of situation, you can also receive it from self. So this is a self-signaling pathway, and that is autocrine, right? Received from self. Okay. So, makes perfect sense, right? All this.
The CD8, CD4, the co-receptor
Mm-hmm.
ones.
Yeah.
So the receptor that's coming out of the T cell that's actually receiving antigen from MHC, that's not labeled in that diagram. We're
This-
only labeling the
this
co-action.
This diagram here?
Right,
Yeah.
that one's not labeled in that picture, it's just mentioned, we're only labeling the CD8 or 4.
Exactly. out of all these shapes on this cartoon, what is the T cell receptor that's interacting with MHC? What color is it?
Is that deep?
The deep red. I know there's some colorblind students, so I apologize. But we've got like two orange molecules on the side, on the flanks, and then we've got this deep red sucker in the middle. That's your T cell receptor. Your T cell receptor is receiving this epitope, this little piece of antigen, that's that blue triangle, and then you've got your MHC1. So this is like the primary interaction we've been talking about this whole time. But now we're telling you that it needs either CD4 or CD8 as a antigen-specific activation signal, and it's also confirming that that is MHC, like self-MHC. Yeah, you first. Yeah.
When CD8 is activated, is the APC, is the CD8 cell killing that APC right then and there, or is it telling itself to apoptosis itself?
That's a really good question. Yes, it is most of the time. So the communication is good enough, and there's enough of a if it's an appropriate signal, and it's not missing any of if it's passing through all the right wickets, then yeah, it's one and done. It will release those cytotoxic granzymes to kill or initiate apoptosis in that target cell.
For the CD4 and CD8, when you say they do not express each other, what does that mean?
It means that you only have one or the other.
Okay.
So an undifferentiated T cell that's developing, it's born in the bone marrow and then it's training up in the thymus, right? It's what we would call double negative. So it doesn't have CD4 or CD8. But at a certain point, before it becomes a mature functional T cell, it's got to get a signal to either fully be CD4 or CD8. And then it'll be single positive.
Like it's only one.
It's only going to have CD4 or CD8.
So looking at the example, now it could be either or.
Yeah.
Do you determine which one it is based off of what it ends up doing?
It's the other way around. What it ends up doing is dependent on whether or not it receives signals to differentiate as a CD4 helper cell or a CD8 cytotoxic cell.
Thank you.
And then once it becomes differentiated as a helper cell or a cytotoxic cell, it's never going to go back. It can never retrograde in function. So it undergoes permanent somatic changes that make it express either CD4 or CD8, and that is what it will always be until it dies.
Yeah.
Yep. Okay. So this might look a little scary too, but it's not that bad. It's just showing the differences of some of those surface receptors on T cells versus B cells and a nice split biplane. So let's see what we can identify here. There's a bunch of stuff we didn't talk about. It's not that important that I wouldn't make you commit to memory, but there are some important things on here. So let's see if we can identify some things. Okay, let's look at the T cell. That's on the left side of this. Where is the T cell receptor? Do you see anything that looks like a TCR? Yeah, the thing that's labeled TCR. What did we say about CD3? It's on the surface of what? It's a marker for what? Mature T cells. All mature T cells. So CD4s and CD8s are all going to have CD3 right next to it. Important stuff. Up here we have a blue-shaped L thing. It says CD4 or CD8. One or the other, but not both. CD2, that was on the other slide as well, has a co-stimulatory function. And then here, we mentioned CD28, and we didn't talk about CTLA4, but I'll get into that in just a second. CD28 would be one of those co-stimulatory surface markers. Looking at the B cell, do you see anything that looks like a BCR? Anything labeled BCR? Yeah, it's not labeled, but you can see the antibody right there, right? And what did we say was in part of that complex? CD19, CD21, and CD20, CD20 and CD21. You don't see CD21 here. I don't know why. Maybe that's most of these two immunoglobulin chains, but regardless, there's your BCR right next to it, right next to that cartoon we've been looking at, and then these two other surface markers that are in complex with it. CD40 we talked about. This interacts with a ligand on some of those APCs. It's got its MHC class 2 because its B cells are also antigen presenting cells, remember? So what do all antigen presenting cells have? MHC class 2, so yep, there it is right there. Okay, so we've got like, and then the rest of the stuff I don't really care about. There might be some interesting things like B7 we talked about, like, those are some of the main surface results we talked about. Any questions?
So you said CD2 was important, where was that, where did you talk about CD2?
Oh, I'm sorry, that's not on the slide. It is, not something you have to remember. Yeah, don't worry about that one. Okay. Okay, cool. How do we feel about the surface markers?
Last card.
There are clearly a lot of them. You mentioned some of them. What are the ones that we need to remember, or is it just the ones on the slide?
Yeah, with heavier emphasis on the bolded ones.
When you say we should remember them, ma'am, are you saying just memorize them by some, this picture that we have of this half moving circle thing, or is it just what they do?
I mean, it's kind of the same information, right? This is just telling you it's surface markers, and this is surface markers. So it's just a visual depiction of the same information. Yeah. Okay. I'm seeing some horrified expressions. But that's okay, over the weekend I will get some review questions together and we'll go over this a little bit and make sure that it's copacetic and we're tracking. Yeah, I'm sure it's overwhelming, but these are kind of like bread and butter surface markers for T cells and B cells, and it is important. With this slide, I think I like it because it kind of tells you that all these interactions are necessary in order to have like a good handshake between an antigen presenting cell and a T cell. That a lot goes into that, and the reason why a lot goes into it is because that's like, you know, top tier of your immune system. This is the adaptive immune system. It's good at what it does. It's complicated. It's specific. It's intricate, and this is like the bare minimum. I mean, it's obviously much more complex than this, but yeah. So we'll come back to that. I know it's a lot. We've got 10:41. Let's take a 10-minute break, and then we'll pick up talking about activation and process. So this is probably not organized correctly, but the recognition is yeah, I think that this is so then there are two responses we can have for yourself, either Lewis granulosis versus round two, induce, yeah. Yeah, exactly. It's like a surface, it's like a single other cell. Yes, this is the most generic figure you could possibly imagine, to just demonstrate this is communication, this is how communication goes between himself and somebody. But, we haven't signed any skills with intentions, we don't know what's being done, but something's being done. So it's just not just standing there dumbly. Signals to frequencies, so, transcription factors, regulated, activated, upregulated, activated, genes expressed, do these, whatever that stuff is, that stuff is being done. It's like interceptors on surface. Unless there's an MRI.
I'm more of a minimalistic man.
But that's separate, that's Yeah, you're like, I'm passing the hallway.
Dying boys. Oh really?
Okay, it got super quiet all of a sudden. Alright, A1, this little bit is kind of a nice continuation of what we were just talking about now that we all have perfect clarity on T cells and how they work. We can move on to talking about even further levels of differentiation and how they become like ultimate effectors of whatever they're supposed to be doing. And also a little bit more refinement on that discussion we were talking about the education of T cells and how they need to be before they leave thymus. So, this is a nice little chart that shows you a few things. Don't worry about it too much, but there are some things that I want you to kind of pay attention to and understand with what's going on here as T cells are developing, what they have to do, all the criteria they have to meet. So you can think of whatever analogy you want to think of, but in the thymus, this is sort of a training and education environment, right? So it's like undergrad or K-12 or basic training or whatever, you know, there's instruction happening here. And instead of getting grades, if you don't pass muster, you just get killed. So, like it says here, 95, the attrition rate for thymocytes who don't graduate from thymus school is like 95, 90%, they get killed, right? So a very small percentage of cells actually survive, meaning both positive selection and negative selection. It's a very rigorous education. But it's important, right? It's important because if you have junky, sub-performing, suboptimal T cells, you're not gonna make it as an organism. You know, you're gonna have autoimmune dysfunction, you're gonna have all sorts of dysfunction. So you need to prune out all these flunkies, and this entire process of somatic hypermutation and mRNA splicing that generates that tremendous diversity in TCRs is fraught with problems. So you need to be able to QC those out before they leave. So very quickly, just to orient you to what is happening here, one of the most important early lessons these T cells get, and like here, for example, DN is double negative, so it means they don't have CD4 or CD8 yet. Baby phagocytes. One of the most important primary lessons is making sure that T cell receptor has affinity for self. So it can recognize friendly, right? It's lesson number one, recognize your own self so you don't attack yourself. That's how to avoid autoimmunity. If these cells are too aggressive, they get down selected, they get selected out. Kind of thinking back to that analogy with basic training, how do we want like our drill sergeants to be? What are some of the attributes of like a perfect drill sergeant?
Very caring.
Sensitive, caring.
Natural basis.
Yeah, upholders of standards, nice and strict, scary, maybe a little intimidating, kind of on the far side of the spectrum in terms of rigidity, I don't know. But there is an excess, right? Like what happens if a drill sergeant touches a trainee? That's considered a line too far, right? You need to select out that behavior as well. So there are margins of selection, and that's why that popped in my mind. Okay, so we have a couple different selection. You don't have to worry about what is happening in each of these steps, but you go through different levels of differentiation, and at some point, briefly, those T cells will have both CD4 and CD8, but that is while they're still in training and they're not going to leave school like that. So briefly, they will have CD4 and CD8, and they will also develop their normalized TCR. If those are inadequate, those get deleted out of the population. And then finally, before they leave the thymus, those double positive T cells will fully commit to being either CD8 cytotoxic cells or CD4 helper cells. So then they become SP or single positive cells, one receptor, but they do not leave the thymus with two receptors. And then from there, as they're going about their normal business, they're maturing. They're initially called naive T cells, but as they start encountering antigens, they become mature T cells. And there are ways to further cull that behavior. Deletion can still occur. Some of them will get deleted out of the population just through lack of love. Like there's not enough interaction, you become obsolete. So through lack of use. And that's fine because you're continually producing more T cells in the bone marrow anyway, so that population will always be replenished. So with the CD4 population down here, you see CD4 T cells, the regular ones, and there's also another subset called Treg T cells, T regulatory T cells. So we'll talk about those a little bit as well, but not on this slide. And this slide is basically just in text saying everything I just said. Naive T cells become activated when they have that initial encounter with an antigen-presenting cell, the right antigen on it. Already talked about this binding that occurs between MHC1 on those APCs, specifically with CD8, and then MHC2 with CD4. You need that co-stimulation with a secondary signal, like B7, binding to CD28, one of those surface markers in T cells. And then once you have all of those, that is your full handshake, right? You have that handshake that this is the correct interaction. What happens when you are perfectly equipped to handle the job? You expand your population, you make clones of yourself, you make more of yourself. So, like as the military is modernizing and we're meeting or needing to meet new requirements for a changing battlefield, whatever, we're going to positively select and proliferate different types of technology that we need to meet those challenges, right? That's basically what's happening here. A T cell meets the right antigen, and that particular T cell gets proliferated. We put all of our efforts into clonal expansion from that cell. And then from there, sorry, there is an extra line in here that's completely redundant, but those activated T cells will then become full effector T cells, which means they're either doing helper T cell work or they're doing cytotoxic work. and then a small subset of those, after that pathogen has been cleared, will become memory T cells that hang around for years and years. When we vaccinate people or when you become naturally exposed to a disease, these memory T cells and memory B cells alike, they're both important in the immune response. So with yellow fever vaccine, for example, when we receive that, which a lot of us have in the military before we go overseas, it doesn't only stimulate B cells and antibody production, you also get a cell-mediated immune response as well. And those T cells, we have data that shows that people maintain some of those T cells 75 years after an initial exposure. So the entire life of that person. I don't know if that's exceptional or if that's normal, but that's a hell of an immune response, right? Lifelong protection. And then similarly, smallpox. You know, how many of you have ever been vaccinated against smallpox? Okay, yeah, a couple of you. That's also kind of like a mother of a vaccine, but when you do get a smallpox vaccine, people have been seropositive, they have an immune reaction decades, protection decades after that vaccination. So very long lasting effects.
So why, so smallpox is supposed to be redone every 10 years, I believe.
Smallpoxes? Good.
If you can tell if you're still immune to it, if you don't, well, you know, that's true. So like if you get the hoax 10 years later, nothing happens to you. Or at least not to you.
Yeah, sometimes they can check it by doing a titration on your blood, right? How much
Yeah.
antibodies.
Yeah, if you, like your vaccination record says that you are vaccinated against all these things, that means you should be seropositive and reactive to those things. And if you aren't, then they simply revaccinate you. Like, you know, I keep showing up, like I'm non-reactive to hepatitis B all the time, so I constantly get hepatitis B vaccines. Whatever, it's fine, but yeah, you can always confirm these things, you can confirm that somebody has an immune reaction, immune protection.
Quick question, so with the older population becoming more immunocompromised as they get older,
Yeah.
if they have memory T cells from all the things that they've encountered throughout their life, why are they immunocompromised? Is it because of the size of the thymus and less T cells are being created?
Yeah, that's a big part of it. So the thymus, like I said, the biggest your thymus is ever going to be is when you're a baby, and then it just shrinks over time. And like I said, it increasingly becomes fatty, and it isn't as effective at training T cells. But also going back to the bone marrow before that, the bone marrow that's generating all these cells, those stem cells aren't as active as they once were. So you have fewer amounts of blood cells being produced. You have less training. And then the existing memory cells from that bout with, influenza you had when you were 13, which happened like 85 years ago, those memory cells are gonna be probably depleted. They're not gonna last forever and ever. So eventually those antibody titers will wane and those memory T cells populations will wane. Yeah. But hopefully you stay healthy and your immune system stays in good shape.
I won't live that long, but I appreciate it.
Yeah, I wonder there. Okay, quick point here, clonal expansion driven by IL-2, just a little note to emphasize again that cytokines are important for maintaining and causing proliferation of T cells. And then this cartoon hopefully should be pretty accessible because we've talked about a lot of this stuff, but a couple things. Let's start with this middle figure and what's going on here. This middle figure is representing what should happen when you have a successful handshake. So what that looks like is you have our APC and our T cell. And our T cell has its TCR, that red box in the center, that is meeting up with its MHC cognate receptor. There's a good handoff of a peptide, there's an actual peptide there. And then you have either CD4 or CD8, that's what this thing is on the bottom. It's coming up there and it's touching that MHC receptor on the surface of the antigen presenting cell that's saying, yep, that's correct, you're the right kind of cell. And then you have a co-stimulatory interaction between CD28 on that T cell and B7 on the APC. So a bunch of different things are happening there. You also have, I said before, IL-2 as an autocrine cytokine, autocrine signaling cytokine is important. So that T cell's also gonna be secreting its own cytokines saying, yeah, this is a good handshake. It's a good handshake, it's important. So all that stuff is important, and that is successful activation. That's kind of what you're seeing in this color enhanced reconstruction. This is an immunological synapse, just like the blurb says. But what we see here is that close association between the antigen presenting cell, which is colored in dark green, so that's this thing on the bottom, that's APC, is a dendritic cell, a DC, and at the top, this is our T cell. So look how close that is. These are all those receptor binding interactions that are occurring in that handshake. Like an actual, it's artificially colorized, yes, but that is what that looks like. Okay, let's look at the top one real quick. This is indicating that something has gone awry. We do have a CD4 or CD8 interaction with MHC, so it's not that. But the problem with this is you don't have a co-stimulatory interaction occurring between CD28 and B7 on that APC. So you don't have co-stimulatory factor, that means it's not a good handshake. This T cell isn't going to be properly motivated to do its job. So that's an anergic response. That means it's just not going to effectuate anything. It's not going to be activated properly. And on this bottom picture, instead of CD28, we see a different molecule called CTLA4 that is interacting with B7. But in this interaction, this is an inhibitory reaction. CTLA4 is inhibiting stimulation of this T cell. So in that case, you don't have any activation. So different interactions will cause different effects with T cells. Okay, in terms of subsets of T helper sites in particular, there are a couple ones we need to be aware of. The CD8 cytotoxic cells, there really aren't subpopulations that we need to focus on. They're all just cytotoxic T cells. But with helper cells, they can become, depending on what signals they receive and what kind of pathogen threats they're encountering, they can become Th1 cells, which are specialized at activating macrophages to fight intracellular pathogens. They can become Th2 cells to fight parasites. They become Th17 cells that fight extracellular bacteria and fungi. And it isn't named Th17 because it's less important. That's the sequential order in which it was identified. So it's super important. Obviously, extracellular bacteria. That's probably 30% of this course is learning about extracellular bacteria. And then finally, a fourth type of CD4 helper cell is called that Treg cell that I mentioned before. And this one is important because it suppresses some of those immune responses. It's always important to have that ability to pump the brakes a little bit, and that's what Tregs do. Yes, sir.
So for these CD4 helpers, are they going to be commonly referred to as Th1, Th2, Th17 Treg, or is it going to be CD4, Th1?
Most of the time when we're talking about T cells, oftentimes we'll just say T cells. If we need to get more specific, we'll say CD4 cells. Very rarely in this course, if ever, do I ever talk about down to the subset level, but just know that they do get that specific. And when they differentiate into like Th1, Th2, Th17, and Tregs, that's another one of those permanent things. Like you're never going to retrograde back to an undifferentiated CD4 cell. You're always going to be that particular subset of CD4 cells.
That's coming out of differentiation.
I don't know if that's before or after they leave the thymus. I suspect it happens in the lymph node. Yeah, once they hit the lymph nodes. Yeah, good question. Okay, so we'll talk, there's another slide that will kind of reinforce this point about these different roles for CD4 cells. With the cytotoxic cells, the only thing that's on this slide that I want to emphasize is that there are basically two different killing mechanisms, but they both ultimately result in the same effect. They both just activate different pathways that result in apoptosis. So one of them relies on, it's just like when we're studying natural killer cells, right? Natural killer cells have these same two options available to them, they're just different signals. And one of those, they release cytotoxic granules, and those granules end up transducing pro-apoptosis signals to that target cell. And in this other pathway, there's a death ligand involved, and that induces another pro-apoptosis pathway. same result, and here, scanning electron micrograph showing this kiss of death between a cytotoxic T cell and a target cell that is expressing a peptide on the surface that is saying, I'm infected with a virus, please kill me. Sure enough, it obliges and it releases those either granules or some other signal cascade that triggers apoptosis. Okay, so this slide is basically just saying what I just said. We're looking at these subsets of T cells. Don't worry too much, this is a progenitor cell. This just means alpha beta chain. So the early progenitor T cell will differentiate into a CD4 cell. Those CD4 cells that are undifferentiated in terms of their subset, they will receive different cytokine cues and signals, and then they will become any one of those subsets that we talked about. Tregs, Th1, Th2, Th17. Don't worry about Th9, I'm sure it does something super important, but yeah, don't need to worry about that. And then this is, I think, the last slide. This is the last slide I want to cover today. Very briefly, it's just summarizing everything. We've seen this figure before, showing that if you have any old Joe Schmo cell that's infected with a virus, whether it be a neuron or a hepatocyte or whatever, a glial cell, that cell should be producing MHC1 on its surface. And if that MHC1 before the cell gets obliterated by whatever virus is infecting it, the MHC1 should, if it's functioning properly, be presenting viral peptides on the surface that can be presented to CD8 T cells so they can do their job and send those death signals to this target cell to make sure it gets killed. There's no mechanism for the immune system to go into that cell and kill the virus, right? The only way to do it is just kill the entire infected cell. So, problem solved, killed. Where those APCs are professional antigen presenting cells, the pathway is a little bit different. Because remember, those macrophages and those dendritic cells are actively engulfing and phagocytizing those targets. So they will get presented differently, but ultimately those peptides still end up on this MHC2 complex, and that too gets presented to those CD4 T cells. So I'm saying the same thing over and over again with complete intention to do that, with hope that the repetition will help a little bit, just reinforcing what happens and how this is different for CD4 and CD8 cells. But hopefully this is starting to make a little bit of sense. We'll find out. And on the left, this too is nothing really new. It just kind of summarizes some of these relationships a little bit. With our Tregs, they play that really important but underappreciated role in calming things down and de-escalating. So those yellow arrows, those who don't know, who can't tell if that's yellow, those yellow arrows are basically, say that with great affection, they have that suppressive effect on all the subset populations of T cells. However, each of these T cell subsets, CD4 T cell subsets, are going to have their own amplification processes with their own cells that they need to communicate with. So the Th1 cells, for example, those were targeting intracellular bacteria, right? Intracellular pathogens. So their interactions with macrophages, this APC like a macrophage, they will activate that macrophage, and that in turn that macrophage will present antigen back to that T cell. So it's kind of like a loop of handling those threats. Same with Th2 cells. You guys remember what kind of pathogens those cells deal with? Parasites primarily. So we have a parasite in here somewhere. That Th2 cell is going to be activating a B cell, and that B cell is also an APC. So it's going to be presenting antigen back to that Th2 cell. And when this B cell gets activated, as we'll learn about more next week, an activated B cell will become an antibody secreting plasma cell. With the Th17 subsets of T cells, you guys remember what those things do? Extracellular bacteria and fungi, yeah, so those are going to be inducing inflammatory cells, which makes sense because these are extracellular bacteria and fungi. So inflammation is going to be one of the best ways of getting rid, like recruiting things like neutrophils, for example, recruiting those neutrophils, making sure that they clear those pathogens. And we didn't talk about CD8 cells, but CTL, cytotoxic lymphocyte, it's the same thing as a CD8 positive cell. Just like natural killer cells, both of these have the same mechanism in inducing apoptosis in target cells. Okay, but just because they induce apoptosis doesn't mean they recognize those infected cells the same way. We talked about that a little bit. What makes a natural killer cell an innate cell is that it doesn't have a requirement for specific antigen presentation. The CTL does have that requirement. It does because it has a TCR, a T cell receptor on it. But otherwise, the mechanism of killing is very much similar, reducing apoptosis. Okay, I think I'll leave it at that. It's still early, so last class of the day. Good luck studying for exam tomorrow. I feel bad giving this lecture on the day before an A&P exam, but you'll be fine, and then we'll review a little bit when we come back on Monday. Do think about this stuff over the weekend if you have time, and you do have an exam on Monday too, right? It's unfortunate. Okay, so you probably won't think about it at all. But if you do have questions, let me know. I'll kind of hang around for a little bit today. And I'm sorry, I know it's really thin, dense stuff, but it's fun, right?